Hi, everyone. I'm Jaclyn Taroni. I want to say thanks to the organizers for putting this together and for allowing me to talk about this recently launched project. Really appreciate it and very glad to be here and hear from others. So I work at a childhood cancer data lab. So that is a part of Alex's Lemonade Stand Foundation, which is a nonprofit based in the US that funds pediatric cancer research. And the foundation started the data lab in 2017, essentially with the goal of accelerating data-intensive research in the pediatric cancer research community. And we do that through a variety of things like training and skill building, building tooling, and also making data available and hopefully more usable. So today I'm going to talk about a project called the Open Single Cell Pediatric Cancer Atlas, or Open SCPCA for short. But before I talk about the open part, I want to introduce the SCPCA part, or the Single Cell Pediatric Cancer Atlas part. So in late 2009, the foundation funded 10 investigators to generate single cell or single nuclei RNA-seq data on the 10x Genomics platform. And then that data, once generated, came to my team to uniformly process that and put it in a web portal called the SCPCA portal. And the reason for that is we want to maximize kind of the value of or research potential of the data being generated. Since then, we've opened this to community contributions, where basically, if you already have pediatric cancer relevant data, you can use our pipeline to process it and send us the outputs, and we release it in the portal. So right now, this portal contains over 500 samples from 50 plus pediatric cancer types. It is mostly made up of leukemia samples, but there are also brain and CNS tumor samples, sarcoma samples, and as well as samples from other pediatric solid tumors. And from the name, you might guess that it is primarily single cell and single nuclei RNA-seq data, but we also have some samples that have bulk RNA-seq or spatial transcriptomics, as well as site-seq data. So if these data sets sound interesting to you, I think it's important to emphasize that in this portal, the data are uniformly processed. So we use a Nextflow pipeline that we've built that we've termed SCPCA-NF that's available on GitHub. And we go from the raw data to summarized data from what you can get from the portal, which is single cell experiment and data format that you can download in kind of various states of processing. We also provide reports, so a QC report that tells you a little bit about the sample, as well as a cell type report, which is what I want to talk about in a little more detail as kind of a motivating example for the Open SCPCA project. So we know that our users want or would be helped by cell type annotation. So we apply two reference-based automated cell type assignment methods to data in the portal, single R and cell assign. So we use these to classify cell types, add those annotations to the process object that you can get from the portal, and produce the report I just mentioned. So this has a big problem, at least one big problem, which is these assignments, being reference-based, are kind of only as good as our references are appropriate for the data we're applying them to. And these are primarily coming from the references, primarily coming from normal healthy tissue and often adult tissue. So we know that there are kind of limitations inherent to applying them to pediatric cancer data. So one way to kind of address the limitation is by using two methods, reporting on the comparison. Maybe if they agree, you're more likely to believe it. So, but that's kind of not where we want to stop. We know that we can do more. We can kind of provide better cell type annotations, but there are 50 plus cancer types. So if there's any kind of like reference that we need to build or manual kind of intervention that we need to do, we should open this up to the community rather than having our small team address this ourselves. So that is that question, as well as kind of open questions that we know exist in applying single cell analyses to the pediatric cancer context, motivated us to start OpenSCPCA, which is a project to collaboratively analyze the summarized data in the open. And we're going to specifically do this on GitHub through using that as a discussion forum, as well as tracking the work of the project, as well as collaboratively managing our code base using that platform. So our goals, we want to characterize the data. Again, an example would be kind of labeling these cell types or cell states. We want to also, along the way, build consensus around the strengths and pitfalls of different methods and really their application in this particular context. And ideally, we'll also, along the way, improve the utility of the SCPCA data to the community, perhaps by coming up with new data assets. So how it works at a very high level is that contributors come in and co-develop analysis ideas with the data lab team or other contributors with relevant expertise. Once we kind of discuss that at a high level, it's off to the races, time to write analytical code, with the data lab kind of helping manage things like software environments, which is important kind of over the lifespan of a project that will likely take multiple years. All code in this will be reviewed in a process called analytical code review. So data lab team members will review code for things like scientific integrity and correctness, of course, but also things like reproducibility. You know, are you setting a seed where you need to? And also things like how generalizable is this code? Can we make it more modular and things like that? And once improved, your analysis is part of the code base, part of OpenSCPCA. Just in more detail, you can either propose an analysis or pick up one that we already have planned. Once that's kind of decided, it's time to implement this. So one thing I want to point out here is if people are interested in contributing to the project, we, in addition to kind of your local setup being available to you, we have we can have you access Amazon Web Services and have a small monthly budget. So we expect an individual analysis to kind of be implemented in stages and kind of have code review and feedback as part of this process. So if that seems like it's more work than just performing analyses, I agree with you. So why are we doing it this way? So first, necessarily, that means that our analyses as part of this project are going to be recorded, which I think is kind of a minimum best practice, scientific best practice. But because things are part of this project and are available in near real time, that means they're out in the world for other people to use and see and evaluate. Again, using cell type labels as an example, they can evaluate whether that is appropriate for their use. And then finally, having this review process built in is one of the better ways that we have available to us to kind of ensure things are more correct than they once were. So that's what everybody gets. What do contributors get? Hopefully access to data sets that they were not aware of previously to help with their science. We're hoping to build a supportive community, have contributors build kind of public analysis portfolios if that's something that would be helpful to them in their career. And ultimately, we're looking to contribute to impactful research. So if you're interested in participating or you know a friend who might be interested in participating, we think the best place to get started is at our docs, but you can also browse our GitHub repos or browse the data itself at the port. I think I'm right at 9 minutes, so I'll stop there.
Thank you very much. Is there any question from the audience for Jaclyn?
Quick question: do you have access? Do you maintain the samples like a biosample repository, or once they're processed, where do they get processed? Where are the samples?
So they get processed at the individual labs.
Okay.
So then the data is what comes to our group, and then we essentially only maintain the summarized data and report.
Right. Is there any feed, like if I work in a cancer center, we have a biosample repository, and so people go and get samples that have various amounts of data attached to them. Do you connect users back to the institutions, like if there's a particular sample that might have had some interesting properties, if they, they could make possibly an exchange of material.
So that is not something that we've come up and kind of explicitly done, but we do list contact information for every single one of these projects, which could be helpful for something like that. But it would be something we would be open to facilitating if it came up.